Okay, guys, so thank you. Congratulations, you got through the first test. I honestly hope everybody was listening when I said how you should use this first test. If you're not happy with your grade, that's all right. But as long as you understood where your strengths, where your weaknesses were going forward. In clinical programs that have exams like NCLEX exams and the ASCP exams for pathology, those types of exams, the tests that you do to practice, that's what they do. They give you where you were weak, and they continue to practice you on where you're weak. And that's not done to hurt you, you know, although it sort of feels like it at the time. It's so that you know what to do to get better. So that's what this should be used for, okay? Okay. Let's go ahead and we're going to move forward. I would really, I want to give you guys a heads up that the next test, because they're going to start coming now faster, the next test will be on chapters 4, 5, and 6. So we finished 4. We're going to finish 5 and probably 6 this week. So your next test will be next week. It will be next Thursday. Okay? And it will be on 4, 5, and 6. This is a condensed course, so we're moving forward. Is it going to be any different than this test? Are all the questions going to be from what we talk about or deeper insights? Yes. So if you hang with me, you know, you'll be all right. All right. So, and we figure it out. But here we go. I've also got another little bit of news. After histology, which we know means the study of tissues, after this chapter, we start organ systems. And what I tend to find is that students feel like all of a sudden they're in familiar territory. And it becomes a little bit, for many students, it becomes a little bit easier. I don't want anybody to slack up at that time because we're going to start, in this semester, we do the nervous system, which is intense. And we also learn all the bones and muscles. So you still have a lot of time management that's going to be important for you to do to get through this semester. But people feel like the information is just a little bit more comfortable for them, right? So hang on. One more chapter, then we get to the skin. Okay? All right. So histology. We've looked at what cells are made of, the molecules that make up the organelles that make up these cells. Now we're going to look at what these cells come together to form, and that's tissues, right? So these tissues, there are 4 major tissue categories, and I'm going to want you to know them. And I'm going to want you to know the basic functions of each of them so that if you saw functions or if you saw specifications, you would know exactly which of the 4 tissue types we were referring to. Do you think I do that because I'm so nerdy? I don't. Let me tell you this. We learned this because there's some diseases that attack specific tissue types. So when you learn about the disease and you learn that it's a disease of the epithelial tissue, or you learn that it's a disease, a muscular disorder, then you will kind of understand what's being lost, right? So this is why we really do need to have this basic understanding. So these are the 4 tissue types, and we already knew from today that 2 or more of these tissue types come together to form what? And that's where we start on the next chapter. But before we get there, let's go ahead and let's take a look at each of these 4 tissue types, and let's learn their basic functions and why they're unique. What characteristics put them in one of these 4 categories. Kind of before we do that, and I do want to give you a little background before we do that, there's some terminology, and I guess it's really all that it is kind of some terminology about this. We talk about extracellular substances, extracellular fluid, and that means what's on the what? Outside of the cell. And then we talk about intracellular substances, right? So you guys have heard me talk about that. For some of these tissues, there's an abundant extracellular amount of fluid. So some of these tissue types have a bunch of that. Some of them have almost none, but some of them have a lot. And you will sometimes hear about that extracellular substance as a matrix or ground substance or interstitial fluids. So I just want you to know when you're hearing those terms, you won't really hear about them too much from me. I'll sometimes say interstitial fluid. But anyway, you will hear about them, and you'll just know that means what's on the outside of the cells, right? Is knowing what should be out there, that it's out there is a right amount, going to be important? Yeah, I mean, it does have to be right, you know, or it could affect how the cells are functioning. But yeah, that's all that's talking about. There was recently, just last fall, they were talking about, anatomists were talking about a new organ that had been, and did anybody remember this from last fall? A new organ, so they were so excited about this new organ that had been discovered in the human body, because we kind of think about anatomy as being a dead science. You know, we have picked apart and dissected apart every part and labeled so much that, you know, we just can't do anymore, except that they thought they found something else. But really, and I don't care if they call it an organ or not, but what they were talking about were the interstitial spaces and fluids, because there's some organs, there's some tissues and organs that have a lot of interstitial fluids, and they were kind of calling that fluid, that space, as another organ, if you will. I don't know, but I think that's semantics. I don't really care about it. But anyway, just so you kind of know that that's what that's referring to. Now there are, did we start off with 4 tissue types? We did? You, when the sperm met the egg, you were 4 tissue types. Or were you one cell? You were one cell. You weren't a tissue. You weren't even a tissue for a while. You were actually what's called a blastocyst. A blastocyst. Guys, what does the word C-Y-S-T mean? Cyst. What is a cyst? No, cyte is a cell. What's a cyst? Okay, so I'm going to tell you what a cyst is. A cyst is a fluid-filled sac. A fluid-filled sac. And we started as one cell, a zygote, and then that cell became 2, and the 2 became 4. That was mitosis that was going on for that. And those cells were in a fluid-filled sac, membrane-bound sac. For the first 2 weeks of development, even after it's gotten to the uterus and implanted, it is a fluid-filled sac of cells, hundreds of cells in a fluid-filled sac. It's a blastocyst. Blastocyst. By the way, if you ever hear the word blast, B-L-A-S-T, blast, it means an early cell. Early cell, if it's referring to a cell, or an early group of cells. So it's very early. So blastocyst, this is very early, right? But then what happens, what will happen, is that 3 germ layers formed. And this is in utero that this is actually happening. And those 3 germ layers, and by the way, I've had students say germ, like bad, like a bacteria. No, which is how people use the word germ, don't they? But let me tell you what germ just means, though. Germ just means actively dividing cells. That's what it really means. Germs are actively dividing. So these 3 primary germ layers, the ecto, meaning outer, endo, inner, meso, middle. Would those prefixes always mean that? Yes. So if you learn them now, that's great, because going forward it'll help you. But anyway, I do want you to know the basics of these. These form pretty early on, and the ecto is going to give rise to the epidermis, which is our skin, and nervous system. You guys have all seen a developing embryo, haven't you, on the Discovery Channel? Haven't you all seen it? And you know, and when we think about in the animal kingdom, all of them look exactly alike, don't they? Really, they almost all look exactly alike up to a point, and then there starts being these differences, right, as they're differentiating. But haven't you all seen where the entire brain is out and the spinal cord? Y'all have seen that, haven't you? And then it closes over, doesn't it? So this ectoderm, really important, skin and nervous system. The endoderm are going to give rise to like digestive, respiratory, some glands. And then the mesoderm is going to give rise to what we call, you know, this connective and muscle. So bone, blood, muscle. So just some basics, know about those. And what this means to you is that the cells, the early cells, the stem cells that are in this area, once they are in these germ layers, is there any going back for them and becoming something else? Or are they kind of dictating their path? And what that really means is that the DNA recipe book is opening up to a particular area, isn't it? So they're early stem cells, but they are, you know, they are differentiating into what they can potentially become. And there won't be any jumping around. You get what I mean? All right. All right, now I want to tell you about something that is, that most people can have seen a boiled egg, haven't you? I want you to think about a boiled egg, like you're seeing right here. I want you to think about a boiled egg and imagine that being a cell. Because aren't our cells three-dimensional? Aren't they, whether they are cubed or whether they are squamous or whatever shape they might be, aren't they three-dimensional? So, okay, so we're looking at a cell right here, a cell that's three-dimensional. And I want you to realize that when somebody is doing a tissue prep and pathology to look at the tissues, a histology slide, our cells aren't purple and our cells aren't green and our cells aren't whatever. So I've had students look at the slides and say, I didn't know our cells looked like these colors. But that's the stain, right? They have to be stained. Now, also, what you've got to keep in mind, this seems intuitive once you look at this, but at first it isn't, is that when somebody is taking a piece of tissue from a biopsy and they cut the tissue, they're sending it down to pathology, histology. And a lot of times the patient's going to stay out, they're going to be waiting up by the phone, waiting to hear what the pathology says. Now, when they take that tissue out, you can imagine tissue, because most of us, you know what that sort of tissues look like. When they get it in pathology, they're going to quick freeze it, and I mean quick freeze it with liquid nitrogen, because they wouldn't want to be cutting on it. Because if they were cutting on it and it was soft, what would be happening to the cells? They would be shredded all over the place. They quick freeze it. They use a microtome, very short knife, and they slice sections that are 1 to 2 cell layers thick. 1 to 2 cell layers thick. They quickly stain it and they look at it. Now they're ready to look at it, right? And that doesn't take long. The whole process doesn't take long at all. Why 1 to 2 cell layers thick? Because the light coming up from the base of the skull, the light has to be able to get through it, doesn't it? If it's too thick, would the light be able to get through it? Would you be able, when you're fine focusing, to be able to see any detail of that 3 dimension? No, because the light's got to get to it. So that's what's being done. Now, wait a minute. So you take a biopsy, okay. So you take this biopsy, you take this tissue, and wherever you put the tissue, there's going to be blood supply, so there'll be capillaries going to that tissue. There's lymphatic vessels in that tissue. There's hopefully tissue from the site you're taking it from, wherever that site might be, whether it's liver or whether it's breast tissue or whatever it might be. And you've got those cells as well. Now, when they slice it, does it make sense to you that if this cell, all of these cells that you are looking at are this cell, but if they sliced it right here, this is the way this cell will look to you. If they made the slice right here, this is the way this cell, does that make sense to you? So what I'm getting at is this: if there was a duct, we wouldn't see the whole duct, would we? Looking at it, we would see something that looked like this. And now I want you to know that when you see a hollow space, whether it is a hollow space like this, or whether it is a hollow space like this, do you see if the duct got cut this way, that this is what you see? If the duct got cut right here, this is what you see. But are all of these the same thing? They are, and your brain is telling you that because you understand that. And here's the thing. The empty space is called a lumen, L-U-M-E-N. So if you ever hear me refer to the lumen of a tube, of a vessel, you'll just know that I mean the what? I just mean the space inside of it. Now look, do vessels have spaces inside just to have spaces, or are they usually transporting something? So they're usually supposed to be transporting something. It's lumen, L-U-M-E-N. So a lumen is that space in between. And you all are going to see some things like that. So these are illustrations that have been drawn for you. And we understand that sometimes there's going to be cross sections here, sometimes oblique at an angle, sometimes longitudinal. Well, I mean, we kind of get that. It's just however those human tissues were done. And so, well, I'm trying to get to a real slide, like something that's real. And I don't really like that one. But does this make sense to you? That now this is actually a human slide, but they have enhanced it by drawing and intensifying these cells. But what would this be referring to? What would this be? We know. These are the lumens. Some of them have been cut this way longitudinally. Some of them have been cut transversely, right? So if they were cut this way, this is the way it would look. If it was cut this way, right, this is the way this looked. So this is actually the slide, and then they enhance this a little bit for you. Do we expect to see in each of these tiny little cube-like cells, do you see each of the tiny little cube-like cells that have a big nucleus inside of them? Do we expect to see a nucleus in every one of them? No, it would just be remember the egg? It would be depending on where it got cut. But we understand that the cell that's beside this one is the same cell. Do you know what I mean? The same. We're going to be looking at these slides. Okay, so we're going to be looking at some of these slides, and you guys are going to have to know in a field of view what you're looking for, right? By the way, I feel like I skipped something, before check, but I didn't. All right, look, so by the way, the next lab is going to be you guys looking at tissue cuts, and you have to tell me which tissue type you're looking at, which of the four you're looking at. Whether maybe you don't know yet because I haven't talked to you about them, but hopefully by the time we finish lecture, you're going to feel pretty comfortable, right, about this. So that's your goal here. So let's go ahead and go to the first tissue type now that we've gotten some background information. Okay, we've gotten some background information. And the first type I want to talk to you about is epithelial tissue. I want you all to know that if I say two functions, you would always choose that is from epithelial tissue. And here are your two functions. And put in please, because I'm just giving you this. If epithelial tissue's two major functions are to line structures, and to secrete products. Now, there's some other things that secrete too, but when you hear line and secrete, what tissue type are we talking about? We are talking about epithelium. We are talking about epithelial tissue. We absolutely are. So when we think about epithelial tissue, if something is lining and it has like many layers, this is called stratified. You all know probably that stratification means layering, right? That's you're talking about the layers. So stratified means layers. Sometimes it's many layers, like your skin, right? And what do you know that is doing for you? What do you know these stratified layers are doing for you? They're protective, aren't they? They're so protective. So think about just what you are looking at when you look at somebody's skin, that all of these cells are dead. And there's more than 30 layers of dead cells. And does that need to be like that? Yes, it does. Because this is our barrier from keeping the external world, out, and our barrier keeping everything we need in, right? Especially fluids. But anyway, protecting us. So stratification, we can imagine just the anatomy of that tells us that it's probably incredibly protective, isn't it? Okay? All right, now, I want you to know that simple epithelium is one cell layer thick. And we said our cells, what's the keeping the outside out and the inside in? What is that structure that's doing that? The plasma membrane is doing that. And we said we know it's mostly made of lipids and proteins helping it to carry out its functions. But one cell layer thick, is that protected? No, that's not going to be protective, but it's that way for a reason. And I want you all, can you imagine what that reason might be? We said it's going to line, so it means that the cells, are the cells going to be real far apart, or do you think they're going to need to be up close together? So in epithelial, do you see a lot of extracellular substance, or do you see almost none? None. So these cells, epithelial, are tightly packed because they line. They're tightly packed. They have very little extracellular substance. They do have some, but they have very little. And epithelial, let me tell you this, do y'all see any kind of, even though it's a drawing, but do you see any kind of blood supply in there? How would blood vessels even get in there? Because they're really what? They're tightly packed. So epithelial tissue is avascular. What the heck? What does avascular mean? No vessels. So wait a minute. If that's the case, that means that where this epithelial tissue is, it's sitting on this membrane here. It's called the basement membrane. You don't need to know that. But it's sitting on this. There must be blood supply coming to this. And how do you think nutrients get to the epithelial tissue? What are they having to do? They're solute, so they're having to do what? Diffuse. And also they're having to diffuse, aren't they? So they're diffusing out. And certainly the ones on this base layer are getting a lot of the nutrients, right? But as mitosis is occurring and cells are getting pushed up closer, further away, but closer to the surface, the cells look, this one looks different from this one, but is it the same cell? It is, but it's getting further away and it's just looking different. Its anatomy's different because it's going to have a different function. And as soon as it gets far enough from nutrient sources, guess what they do? Well, yeah, that's called dander or exfoliation, right? But what are they doing? They're dying, but is that a good thing? Yeah, they're dying because they're forming this barrier, right? So in skin, that's what they're doing. Like the inside of your cheeks, right? The inside of your cheeks, do you think that's simple or do you think that's stratified? That's stratified, folks. Simple is not going to be protective, is it? How about linings like in your intestines and or vaginas, ladies? What is that? That's stratified after puberty. Before puberty, it's not. And that's why it's unnatural and not recommended that vagina is not a birth canal until after puberty. Right? Because before puberty, the epithelial is very different. And this is why sometimes you'll see little girls come in that have been abused that will bleed out on you before they get to you in your emergency rooms. There's no protection. So anyway, so simple we're saying simple is one layer, and we know it's tightly packed, but man, that's not seeming very protective. So what would be a function of that? Why would we want something that had just one layer lining? What'd you say? Does somebody have a guess? What'd you say? Brad, come on. Right on. You said. I can't hear though.
You know how the first layer is shit.
No, that's all dead. And so that's no, but I'm talking about an actual structure that these are living cells, living, and they're only lining something with one layer. One layer. Guys, would it make sense to you that structures that are going to be in areas where rapid absorption between the two spaces has to happen? So gases from your lungs and your little microscopic alveoli, the oxygen you just were breathing in has to quickly get across to the blood, right? And CO2 has to quickly be released back. Hey, how about those capillaries that's down here in the alveoli? So you've got this little microscopic air pocket called the alveoli in the lungs. And this microscopic air pocket is lined with single layer epithelial cells, alveolar cells. And underneath that are blood capillaries. And guess what? Guess what blood capillaries? Well, they're simple. They are single layer as well, because do you have to have a rapid diffusion and happening between them? You do. So where we need rapid absorption, this is perfect design. This is a perfect design. That's why people should be careful with their alveoli, right? And one of the reasons. But okay, and capillaries, blood capillaries have the same thing. So, anyway, I do want you, we're not going to look at all of the epithelial tissue types, but we are going to look at simple cuboidal, and cuboidal means that they're shaped like what? They're shaped like cubes, and we're going to look at simple columnar, and columnar are shaped, these are cubes, right? Little cubes here. And we're going to look at simple columnar. Simple columnar, these columns, they look like columns here, and simple because they're one layer. It's just we don't have enough time in histology to look at every tissue type, but these would be two that you all will look at when you're in the lab, and we're going to take a look at them. When we look at these, and this is what the real slide would look like that had been stained. This has been enhanced, illustrated for you. But here's the nucleus. Do you see the nucleus? Here's a nucleus. Here's a nucleus. Of course, they got cut in different spaces so they can look kind of funny, right? Some of them might not even look like it had a nucleus, but did it? Yeah, just we know where it got sliced. We just didn't see that. But I do want you to know that there's also a special kind of cell that belongs to epithelial tissue called goblet cells. Goblet cells. And they're sometimes referred to as goblet cells because what does this look like a little bit? Kind of. I don't know. Whatever. I'm upside down. Okay, but whatever. But what this goblet cell is going to release is mucus. A goblet cell produces mucus. Is mucus a good thing or a bad thing? It's a good thing, isn't it? We said that it's a really good thing to have mucus out here because we also have chloride pumps that are working that's keeping that mucus nice and thin, right? And we have maybe either microvilli, in some areas we have cilia that are keeping things moving along. But mucus is actually a good thing. So we do want to know about goblets and whatnot. Do you have any questions for me about epithelium, epithelial tissue? So we said it's going to protect. We said it is going to, we said it's going to protect. We said it's going to line. It's going to be secreting. So it's going to be secreting products like mucus, but there's also others like endocrine glands that secrete. So that's their epithelial. And we know that these secretions, sweat glands, right? So all those things, those secretions are going to be important, aren't they? All right. For us to have. Now, I'm going to move through. If I move through it, are you responsible for it? No, we're going to get to the skin next, so we'll go through that. I didn't know I had so many slides about epithelial. Are you kidding me? Okay. All right. Now, I think I told you what I want you to basically know about epithelial tissue. And you all know that we said when we get to the organs, they're going to be composed of 2 or more of these tissue types. Most organs have all 4 of these tissue types. But to be called an organ, it's going to have 2 or more, and that's why I don't think interstitial things or fluids are going to be organs. But anyway, I don't care. I don't care if they name them an organ. So now I want to give you the next one. And the next one, let me say this to you guys about this next one. This next one, the next one is the most abundant tissue type, connective tissues. They're the most abundant and they're the most diverse in what they look like. We know something's epithelial if the cells are really what? Packed tight. They're really bound tightly together. But these connective, we're going to see all kinds of tissues in this, but they do have some basic things in common. Connective tissue, most of them are highly vascular. So, going to be highly vascular. Widespread blood vessels are going to be abundant and all of that. And what we know is there's going to be a lot of extracellular substance, a lot of extracellular matrix, a lot of ground substance, right? So there's going to be a lot of that. Certainly, there's also going to be cells that make up tissues, because we know cells make up tissues. But in connective tissue, there's a ton of extracellular. substance. I want to give you some examples right now, and it's probably on the slide, but anyway, some examples are going to be blood. It's hard to think about blood because, as a tissue because it's liquid, but it is. It's a connective tissue. And then I also want you to know that bone is a connective tissue. Bone is a living, dynamic tissue. A lot of people think about bone as being, you know, you think about a bone and you think, how can that be alive? You know how it's alive? Because the extracellular substance is crystallized and it looks hard. But guess what? The cell's alive. If bone loses blood supply, it dies. And that is not a good thing, right? So bone is a living dynamic tissue, has a ton of extracellular matrix, and it's a connective tissue. And we're going to find when we get to that chapter about bones, all the functions that bones have, more than I think you know, coming in here.
Why is bone, well, if they're living, why do they last longer than the skin and organs?
Last longer?
Like when someone dies.
Oh,
why is the bones?
Oh, you mean why are they the first? Because of that hardened, 2/3 of bone is this hardened extracellular matrix that's crystallized. But the cells die when this, you know, when someone dies, not all cells die at the same time. They don't. So they don't all die at the same time. I mean, we have a clinical definition for death, but not everything's going at the same time. But if you're asking like why they don't decompose as quickly, it's just because of that hardened matrix. And we're going to talk about what that matrix is made of. They're calcium complexes that make it so hard looking. But bone osteocytes are alive. They're living tissue and they're dynamic throughout your life, even if you live old. They're living tissues. Okay, so we think about this as being highly vascular. And then some other adipose. Adipose is what do we call adipose tissue? Fat, right? So we call it fat. Adipose, blood, bone, cartilage. Cartilage is a little bit of an exception to this highly vascular. It's the one connective tissue that isn't highly vascular. Still has a lot of extracellular matrix, but it's not highly vascular. This is why if somebody has injury to cartilage, it can take a really long time to heal. Have y'all ever heard of joint injuries being so much longer to heal than a broken bone? Mm-hmm. And that's why, because joints are filled with what? Cartilage and tendons and that kind of stuff. So, okay. So we know that there's going to be a big variety. This is going to be our biggest group. So diseases that attack connective tissues like lupus, y'all have all heard of lupus, haven't you? Diseases that can attack connective tissues, we need to understand what that patient might be losing. So some of the things they have in common, binding organs together. Y'all have all heard of tendons and ligaments. And I want you to know the difference between a tendon versus a ligament. A tendon is holding a muscle, which is a different kind of tissue, a muscle to bone. So it's connecting a tissue called muscle to a connective tissue called bone. So tendons connect muscle to bones. What do ligaments connect? Ligaments are connecting bone to bone. So if you think about tendons that are connecting muscles that actually, move across between two different bones, right? There's something at this joint. Can you imagine the instability of this joint you had if you didn't have ligaments stabilizing it? So ligaments are these connective tissues that are stabilizing two different bones at that joint. Connecting bone to bone. That's a ligament. All right. They are for protection. Certainly our skull protects our brain, right? And our ribs can protect our thoracic organs. A lot of people say chest was pectoral, and people put different things on those flags. But anyway, our thoracic cavity is protected by the sternum and the ribs and these bones, and we get that. And then when we talked about blood, you all know that blood, or maybe you did, but blood is mostly liquid. It's mostly extracellular fluid called plasma. More than 50% is plasma, which is extracellular. And of the plasma, that's mostly water. But inside of that blood is also part of whole blood is going to be cells. And you all know that cells can be red cells, white cells, or platelets. Have you heard of those three? Okay, so at this point, I would want you to know that the white blood cells, they're also more accurately referred to as leukocytes. Leuko, L-E-U-K-O. You can do it with a C. That's accepted too, just European versus American spelling. But leukocyte, one word. Leuko as a prefix always means white. Always means white. Erythrocytes are what kind of cells? Erythrocytes. Red, because erythro always means what? Red. That's what it means. So when we learn this terminology, we learn it as we go, because will this help us going forward? Yes. So anyway, looking at these white cells, they protect us. They protect us. We live in a microbial world, don't we? But we're not sick every day of our life, are we? If we were sick every day of our life, we wouldn't make it out of toddlerhood. Because if we've been sick the entire time, we would have just succumbed. But we're not. We live in an ecosystem that's filled with normal flora, filled with potential pathogens. But we're not sick all the time. And actually, if we have strong immune systems, we're rarely sick. Is that right? All right. Movement. You know, it's hard for you to think about movement and think that bones have something to do with that, but they absolutely do. A muscle attached to two bones is why you have movement. It is why you have movement. If I were to like dissolve or suck all of your bones out of your body right now, you would just be a little quivering jelly mess if all you had were muscles. But because you had a muscle, one muscle's skeletal muscle is attached to two different bones, when the muscle contracts, what's going to have to give? Those two bones it's attached to, so then you get what? Movement. Right. Has anybody had a kinesiology course? Right. So I'm sort of setting you up for this idea about bones and muscles and giving you some basic information. And will that help you when we get to those chapters? It should. Okay. And then storage, fat. We said you have to have enough fat, don't you? Actually, adipose you have to have an adequate amount of adipose at puberty to even start reproductive cycles effectively, right? Women have more adipose than men, and that's normal. They're supposed to, because women have the gestation periods, right? And humans have usually gone through periods of famine. So women's hormones have designed to be beautiful at helping them to conserve calories, extra calories that are taken in for that reason. But anyway, adipose is also important. So we think about that obviously in reproduction, you know, that women have to have enough of that so that the developing embryo, fetus, that has what it needs. But it's for other reasons too. You have to have enough layer on because it's under our skin in the hypodermal area. So it insulates, it protects. And then we also have enough that it can stabilize certain organs, or some organs, if you drop below a certain body fat content, the organs aren't stable at that point. So you can get kidneys torquing a little bit. And you also need it for alternate fuel sources. We said that that's a good alternate fuel source, isn't it? What's your best fuel source? Carbs. But fats can be too. So heat, the brown fat that's in babies, this can help with that. Babies really can't thermoregulate the way that we can. If we get hot, we can sweat, can't we? We can shiver, we'll get signals for that. And if everything's working well, then we do it perfectly. Sometimes we got to be consciously knowing it. And behaviorally, we can take things off or put things on if we're cold or hot. But babies actually can't thermoregulate the way that humans can. It's because of their surface ratio is off, and just their kidneys are young too. So you have to be really careful. How many of y'all have seen babies out that you know darn well they're too cold, but they're not being or too hot? Mm-hmm. So the adults treating the baby like they feel, but they really need to be more aware of ambient air temperatures for babies because they really can't regulate as well. Okay, so all right. So these are just some of the cells that make up connective tissue, and I am going to give y'all a break in just a second. But let's do these cells first because I know that these can seem a little overwhelming. I talked about adipose as tissue, isn't it? Fat tissue, and it's composed of cells called adipocytes, which are what? Fat cells. That's what they're composed of. I also have maybe talked to you about, well, I haven't, but I will now. I'm going to talk to you about something called fibrocytes. Fibrocytes, they make up these fibroblasts, make up fibrocytes that end up being these fibers that we find in a lot of connective tissues. Have y'all heard of collagen? Wish I could find some. Have you heard of elastin? Wish I could find some of that too. Elastin, collagen. These are all incredibly supportive fibers that help to keep the integrity of these tissues, doing what they're supposed to be doing. And then I want you to know this word macrophage. I already told you what phage is. What does phage mean? To eat. So a big eater. Macrophages are made of special kind of leukocytes called monocytes. And we have these in most of our tissues throughout the body. We have these monocytes that end up becoming macrophages, that I call your big dump trucks of your system. Monocytes are in really every place, because don't you have cells that are recycling themselves? Don't you have waste products? Don't you have injury to things? You could sneeze hard and have bleeding, in some of these tissue spaces. But anyway, it all gets cleaned up, doesn't it? What helps to clean it up? The dump trucks. What are the dump trucks? That become macrophages. That's exactly right. And then we have other kinds of white blood cells too, of the leukocytes. Neutrophils are special kind of white blood cells that protect us against bacteria. Neutrophils. And lymphocytes protect us against, and this is, you need to take this out because this really needs to come out right here on this slide. This is from your text, but it needs to come out. Lymphocytes protect against viruses, and cancer. Every day of our life, there are cells that lose their cell cycle control mechanisms. But it's okay because we have these lymphocytes that take them out, that will destroy them. I told you all that you have this glycocalyx layer where your tissue proteins or self-identity proteins are, and that you have cells that are just looking for ones that are foreign. And if they're foreign, what are those cells going to do? Kill them. Isn't that right? We have what are called natural killer lymphocytes. That's all their job is to look for something that has lost its life cycle mechanism or has foreign proteins, and it destroys them. These things are good. They're no joke, these cells. This is why we can get out of infancy without having cancer, right? And or having a virus kill us. Because there are viruses everywhere. We live with viruses. Most viruses don't even keep a child from skipping and jumping. And
does that lymph nodes get enlarged in?
Well,
when you find.
So we'll talk about lymph nodes when we get to the lymphatic system, but I'm glad you're thinking that, because we do have these nodes that filter lymph, and they're filled with these lymphocytes, and they are. So if they're ever enlarged, it means that they're actually what?
It's a good thing.
They're working.
Okay.
They're working. That's not a bad thing. Can they get too big?
Yeah.
Yeah, and they can sometimes hurt, and it can take a long time for it to drain. But we'll talk about that in a moment. But yeah, so good thinking about that. But these are cells that are part of that system. And we have a lot of different kinds of lymphocytes. One of the kinds are natural killer cells. They protect us against anything that's foreign, and cancer cells and viruses. It's viruses I want you to know about. And then, guys, have y'all ever heard of antibodies? Have you ever heard of their proteins? And I want you to know their proteins. I want you to know their proteins. And have you ever heard somebody say, oh, I can't get that because I've had that before? Have you ever heard somebody say that? What they were really saying was, if it's true that they had it, they made these proteins called antibodies, and they won't ever get that again, that same thing again, because these antibodies are proteins that are specific, that are going to fight those specific bacteria or pathogens, and that you won't get it again. That's the way your immune system's supposed to work, okay? So, and it's really true. We have the ability to make millions of different types of these antibodies, and they are proteins that are going to be protected.
Like chickenpox.
Yeah. So if you've ever heard that, you know, you had chickenpox so you can't get it again, it's true. Now, wait a minute. That doesn't mean your mama didn't say you had it twice. So if somebody's mama said, oh, no, she had chickenpox twice. Mamas are usually right, but they're wrong about that. Because let me just tell you, there's some things that can look like chickenpox that aren't chickenpox. So unless somebody got tested, they might have thought they had chickenpox twice. But that is one of the ones that you don't get it twice. You really don't. Now, immunizations are a little bit different, but in a natural setting, getting natural disease, there's some that you're going to get and you won't get them twice. You won't. Now, once you've had chickenpox, it's a virus, so can it live in your system and come out later? What does it come out as? Shingles. Yes. So there are many viruses that do that. They can stay in our systems and be latent. That means they're just resting. They're dormant. They're resting. And they don't all, not everybody who had chickenpox will get shingles. But about a third of the people who had chickenpox will get shingles. It'll come out later. Anyway, any questions about that? I'm trying to give just enough information that doesn't make it so confusing at this point. But those antibodies are circulating around, and they're in our tears, and they're in our saliva, and they're in our synovial fluid, and they're in our blood. And they're protected, aren't they? Because if you get exposed to something that second time, you don't have to be laid up with it for that time. I don't remember having chickenpox, but I was told I had chickenpox, and I'm pretty darn sure I did, because when I had a 1, 2, and a 3-year-old, actually did 6 months, 14 months, and whatever the other one was, no, 15 months, whatever. They were all under 3, okay? They all got chickenpox at the same time. So So I was inhaling chickenpox virus, swallowing chickenpox virus, rubbing it in my eyes. But did I get sick? Why not? And what did I have? I had antibodies. So as soon as I started inhaling and wiping, and this is just so many pops. So anyway, my antibodies took them out. And I never got sick again. You don't get it, right? So anyway, so that's antibodies. And we have those against anything that was foreign that we've ever encountered in our system. And we live in a foreign ecosystem, don't we? But that's okay, because we have the ability to do this. Yes?
When they say, don't they say that there's antibodies in breast milk as well?
Oh, it is.
So, so when you breastfeed, like, for example, the chickenpox, would they still get the chickenpox if they were exposed to the virus?
That's a great question. So when you are breastfeeding, and it's one of the reasons they highly recommend people to breastfeed, if you can. But if you can't, you shouldn't, because it's not about you. It's about your baby getting what it needs. But I've known some women who were just so anxious about breastfeeding that they were not maybe paying as close attention to the baby's diapers and whatever. And it's not about the mother; it's about the baby. The baby's not getting what they need, and you gotta give up, or you gotta do something else, because the baby's gotta get what they need. But the breast milk, especially the colostrum, that first milk, is filled with antibodies, the mother's antibodies, that will line the baby's GI tract, and that really protects that baby. If that baby encounters like even little viruses, because you know everything's new to that baby's immune system. You know what the number one cause of death in infants is? Dehydration. And you know how sad that is? Because that could be, that's a symptom, and you can actually treat that, right? But the babies are very prone to crashing if they don't have the right hydration. So anyway, and they can pick up things. So breastfeeding, even just for a few days, can really line the GI tract with the mother's antibodies. But they don't last in the baby. So they don't last. So soon they break down. They're not the baby's antibodies. They're the mother's that the mother's off, giving, and they won't last for that long. So this is why they say that infants that are breastfed have the mother's protection. But even antibodies that cross the placenta will protect the babies the first few weeks of life, the ones that cross the placenta. But that doesn't mean you should take them to Walmart. Do you know what I mean? Because you shouldn't. They've got baby immune systems, right? They're weak. They're immature. They're not weak. They haven't even gotten strong enough to be weak yet. Do you know what I mean? They're immature immune systems. So, but they have some protection from the mother. What cross the placenta, the antibodies that some can cross the placenta. And if they breastfed, what's in the lining that tract. So that's why they strongly encourage that. Breast milk does. Any of our body fluids have antibodies. All of our body fluids have antibodies circulating around. And again, it's why we don't get sick. All right, so we're going to take, I had told you about collagen and elastin, but we're going to take a quick break. This is solid steel connective tissue. And we're going to come back in about 10 minutes, 15. How much do y'all want? 10? Okay, 10 minutes. I'll see you in 10 minutes. I think y'all were hearing that yesterday the recording didn't have audio, and I'm just keeping my fingers crossed that this recording has audio today. It's just yesterday's class. Y'all, it's a different class, and then it wasn't working at 65 either, but for different reasons. But anyway, so I'm hoping, but just so you know, might not be today. All right, so when we think about like elastin and collagen and those structures, elastin, you might think about elastin as being tissues that are left to stretch, but that's not actually the function really of elastin. Elastin certainly helps with that, but it's so that tissues can rebound. So that's a main function of elastin, is so that the recoil, it does allow the stretch, but recoil is really important too. So when we think about tissues that need to be able to stretch and then rebound, we think about like the tissues that are surrounding vessels and that sort of thing, so that when you have, they also surrounded by muscle layer as well, but so that they can actually come back to the original thing. This is going to be important as the stretching. So these are going to be some of the fibers that we think about. The ground substance, I don't really worry about too much. This is a beautiful dissection of the tendons and whatnot in the hands. And you can see how delicate this dissection was for that. That's just sort of FYI. The tissues that you will look at in histology, it's going to be 3. It's not going to be these, but it's going to be compact bone, a blood film, and adipose tissue. They're going to be the 3 that you all look at for the histology lab. You know, we don't have a whole bunch of time for you to be looking at a lot of these others, like areolar tissue or collagen and that sort of thing, but cartilage. But you can almost guess that this was connective tissue, because do you see how much extracellular space there is in this? You're not seeing cells that are tight together, are you? No. Okay. So anyway, lots of pictures, but I'm going to be very particular about which ones you're going to have. And I'm going to show you. I'm going to actually show you the ones, if I can get to them, of the pictures that you'll have. So that will be one of them. And so this is, sometimes the students feel like this is the hardest one because they're not quite stained this darkly. So what they really look like are almost ghost cells. But these are cells. They look empty, but are they empty? No, they're filled with fat, right? And every now and then you can make out a nucleus if it got cut just right, or sometimes even a blood vessel. But this is what adipose does look like. And then right there. And then I'm not going to have you look at cartilage. I just want you to know cartilage is a connective tissue. we find cartilage at the ends of structures to give support, right? Like this is all cartilage right here. Cartilage between bones to cushion. So we've got cartilage in different areas that we see. And then compact bone looks like this. And so nothing else looks like compact bone, but you guys will be looking at that as well in the lab. And then also blood. So when we look at a blood film and we are looking at like high magnification 45 or 40 objective in there, which is 400 magnification, looking at these fields of view, most of the cells are anucleated cells. What does that mean? Anucleated. Without a nucleus, and that's what these cells are, red blood cells. Some have nucleuses, and the nucleated cells are white blood cells. There are way fewer white blood cells than red blood cells. The cellular component of blood is almost entirely red blood cells. But anyway, and what was this in the background? It's what? It's a fluid called what? What's the fluid part of blood called? Plasma, right? So this is the plasma. This fluid extracellular space is really not empty space. It was plasma that this blood was in. The nucleated cells are white cells, and there are a lot of different kinds. And these little partial cells that you're seeing here that almost look like dirt on the slide, they're not, it's not dirt on the slide. These are platelets. Platelets are partial cells. They only live for about 96 hours in circulation, but they do have an important function, and we'll talk about that. All right, so so we're done with connective tissue, right? All right, so let's look at nerve tissue or nervous tissue, if you will. What I'm going to give you all is just one slide. It's a motor neuron I'm going to get you to look at. These cells are huge. These cells are. This is the nucleus of this motor neuron. And here's another motor neuron. And it looks like these cells have appendages coming off of them. Does it look like that? Mm-hmm. Because these neurons can be more than a meter long. And y'all know a meter is like a little over a yard, right? So these appendages really can be long. This is the body of the neuron. That's the nucleus of the neuron. We know if we had an electron microscope, we could actually look in here and see the organelles, couldn't we? But what I'm going to want you to know for right now is this. I'm going to want you to know there are two major cell types in nerve tissue, and they are neurons and glial cells. Glial cells. Here's the glial cells. You will also see them as neuroglial cells, but you will also sometimes just see glial cells.
What's the first one?
The first one's a neuron. Neurons and glial cells. If you heard that something was a glioma, what would it be? A tumor of the what cells? The glial cells, right? Of these glial cells. So anyway, two cells for now. This is what we're going to learn about the nervous system. Neurons and glial cells. Neurons are these big cells, and this has been illustrated, but sort of looks like what we just saw, the real one, didn't it? This looks like an octopus or something going on here. These neurons of the nervous system get all the glory. They get all the glory because they're doing the detecting from internal environment and external. So they're doing the detecting. They're sending signals throughout the cell to the next cells. So they're communicating. They're detecting environmental stimuli. They're responding to it by talking to the next cell. Sometimes the next cell is another neuron, and that's going to send to the next neuron, and that's to the next neuron. Sometimes these neurons talk to muscles, and if it talks to muscles, what do you think the muscles are going to act on that? Sometimes these neurons are talking to endocrine glands. What do you think the endocrine glands are going to do in response? Produce a what? A hormone. So these neurons, they really are, they kind of should give you glory. They're setting set point ranges. They're detecting when things get high or low. They're detecting stimuli, responding to it by sending signals to the next places it needs to go. They're great communicators. But okay. All right. So, okay, but they are. And so you've got to have them functioning. But here's what I need you to know. The glial cells way outnumber the neurons. They're called the helper cells to the neurons. And they are not, they're the helper cells. And do you think the neurons are going to be able to do what they do if they don't have the glial cells functioning? Mm-hmm. So one of the first things that they come out before I spit it out on sorry about that. I'm back. All right, so no, they can't. And so it's going to be really important that these glial cells also be functioning. And one of the first things we do when we get to the nervous system and we're looking at the nervous system is we'll learn about the 6 glial cells that we know about, the 6 different types. And we're going to learn the functions of each of them. And you think it's going to be important for you to know that when we get there. But for right now, what do you know glial cells do? They help the neurons. They support the neurons. The neurons can't do anything like they should if the glial cells aren't functioning. There's some very specific diseases of specific glial cells. Multiple sclerosis, MS, is a disease of one of these glial cells. Alzheimer's, a disease of one particular function of one of these glial cells. So we want to know about that, don't we? Right? All right, so neurons. So neurons are kind of cool that way. Now, you're realizing that the cells and the tissues that we've talked about, we're in the 3rd major category right now. The cells and tissues that we've talked about have all had very different kind of functions, haven't they? And maybe it would make sense to you that the cells that make up those tissues, they don't all live the same amount of time. I told you that platelets are only going to be there for about 96 hours. Some of those monocytes won't last that long, right? But then we have some cells, like these neurons, that had better last because they don't undergo mitosis very often, especially the ones in the central nervous system, which is the brain and spinal cord. So do these things need to be protected, these neurons? Yeah. And so we'll talk about the peripheral nervous system versus the central nervous system. But all of these differentiated cells, whatever path they've differentiated into, whatever they've become, it's going to be dictated from that, how often they divide, if they can divide, how often they divide, and how long they last. I would hope that kind of makes sense to you, right? Okay, so that's the nervous system for now. We are going to end up having, honestly, we're going to end up having 4 chapters on the nervous system at the end of this session. So we talk a lot more about it. I don't think it's a bad idea to tell you now about this neuron. The body of the neuron is called the soma. If you ever see a prefix neuro, you know you're talking about one tissue type, nervous tissue. But it's usually just referred to as the soma. But anyway, that's the cell body. And then the dendrites, this isn't a great picture of the dendrites, but the dendrites are what I call the feelers. They're the little feelers. They are what are detecting. They're what's detecting the stimuli. So the dendrites, there's got to be a better picture. Okay, anyway, so that's what a dendrite is. And then the axon is going to be after the dendrites feel and detect these ends that are way down here. They're detecting the stimulus. The stimulus will be sent through the body and along this what's called an axon. And that information gets sent to the end of that axon, and then they will release something called a neurotransmitter. That's a chemical. That's a chemical. So neurons release and communicate by releasing neurotransmitters, which is just a molecule. It's a chemical that is going to cause a response in the next cell. What are some of the next cells that we know about? Other neurons, muscles, endocrine glands, so these are how these neurons, just some basic anatomy of those neurons. Are we good? Now muscle, we also have a couple of chapters that we're going to deal with the muscular system, muscles, okay? But what I want you to know now about muscles are some basic things about muscles. And what I want you to know now is that there are 3 types of muscles. 3 types of muscle. There's skeletal, cardiac, and smooth muscle. Skeletal muscle, cardiac muscle, and smooth muscle. So where do you think you find skeletal muscle? Attached to bone. That's exactly right. That's why it's called skeletal muscle, because it's attached to bone. What do you think the function of skeletal muscle is? Movement. That's what it is. It's movement. Skeletal muscle is voluntary. What does that mean?
It doesn't have a choice.
It does have a choice.
So you can control it. You decide to write notes, and you're writing them in a very particular fashion, aren't you? Because you are voluntarily controlling your muscle movement. Just knowing how many muscles that takes, it's actually pretty amazing what's going on for you to just take notes, right? Not to mention turning the cartwheel. All right. All right. Do you know there's a day that's the last day in your life you turn a cartwheel? That's sad. Somebody's looking at me like I'm being serious. But I am being serious in this case, because I don't think I'd ever try to go ever again. So there was that last day, and I didn't even know it was the last day. I would have done one the next day if I'd known that. But then that would have been the last day. Okay, now I'm good. All right, so skeletal. Skeletal muscle is attached to bone. It's under voluntary control. You control what you're doing, right? Now, do some people lose that ability and have involuntary things? Can that cause them problems? Certainly it can. And so when we get to the muscles, we'll talk about this. But anyway, cardiac muscle, where is that found? It's found in your heart. That's your heart is mostly muscle. It really is. When we dissect hearts, people are just amazed at that myocardium, it's called, that muscle that heart really is. Okay? So cardiac muscle. It's slightly different than skeletal muscle. One of the ways that it is different is that when you think of the heart, you've got the chambers of the heart. Is that right? You've got these chambers of the heart. Is that correct? Mm-hmm. The heart is not under body. Are you having to think about every heartbeat? You would not be able to do much else if that's what you were thinking about. But you have a little group of cells that have one function. They start right before the 8th week in embryo. And this little group of cells is called the SA node. It's in the upper right chamber, the upper right atrium. This SA node stands for sinoatrial node, sinoatrial node, SA node, and it is the pacemaker of your heart. It is a little group of cells that start firing. They send an electrical shock through the heart, and it starts firing. And it continues, hopefully, throughout your natural life. This is your built-in pacemaker of the heart. Can things influence the pace of your pacemaker? Mm-hmm. Certainly, because you can get scared. And if you get scared, what happens to your heartbeat? It gets faster. Does somebody say it skips? It can skip. Okay, well, it gets faster, and then you can actually voluntarily slow it down by taking a really deep breath and putting pressure on your thoracic cavity. And we'll talk about what that is called. It's a vasovagal response. But you can do that because then other things like acetylcholine, it's a neurotransmitter, will slow it down. So there are other things that influence it, but is the heart autorhythmic? It's autorhythmic. It's not under voluntary control. Thank goodness we have enough to think about, don't we? Right. And will you know about the SA node? Yes. Smooth muscle. Where is smooth muscle? Where would that be? Smooth muscle is going to be surrounding body organs, such as your 30-foot GI tract. 30 feet. I want y'all to think about 30 feet. When you swallow something, you voluntarily swallow. But then you don't have to think about that food again for 30 feet. That tract has smooth muscle, that GI tract has smooth muscle surrounding those tubes that are moving that substance along. It's very coordinated activity, it should be. And it's coordinated, they're called, you don't need to know, peristaltic contractions, we'll know that when we get there. But these peristaltic contractions are coordinated contractions that are keeping things going in one direction, right? Until 30 feet later, we can voluntarily control getting rid of what couldn't be digested, right? All right, but for 30 feet, you don't have to. How about your blood vessels? Your blood vessels are carrying blood to all the tissues and throughout the body, and they need to be. But you all know that on really hot days, the blood vessels look larger to you, don't they? And on cold days, they look like they're smaller, right? But what has happened? Smooth muscle has gotten signals to either contract or relax, dilate. On cold days, they will relax, blood will come to the surface, it will help to get rid of some of the heat from the core organs and keep your core organs where they need to be. On cold days, they're going to contract down, that smooth muscle will contract. Do you have to think about it? Mm-hmm. Can you even control it? Involuntary control, not under voluntary, not under voluntary. These smooth muscles are mostly working because of communication from the nervous system talking to structures that are going to control this. It's mostly the hormones. So how about your bladder? When you urinate, you're not thinking bladder contract, bladder contract. You're not doing that, are you? Your bladder, which is a fluid-filled sac, your urinary bladder is a fluid-filled sac. That's why cystitis means what? What is cystitis? I told you what a cyst is. Cystitis is inflammation of the urinary bladder because the urinary bladder is a what? It's a fluid-filled sac. What is it usually filled with? Urine. It stores urine between voids, doesn't it? But when you urinate, you don't even realize it, or you should realize it, because people who do realize it means they have something wrong, and it's usually painful. They say it's painful. But you don't realize it, but when you have voluntarily, you're urinating, your bladder is contracting, trying to void all of the urine. Okay?
So the smooth muscle surrounds
body organs, and it surrounds body organs, and the function of smooth muscle is to control organ volume. And can you see how it does that? I mean, you can see that, right? It controls organ volume. We also have muscles that are round that are called sphincters. Sphincters, round muscles, you have a sphincter here, sphincters. So you have sphincters for urinating, internal and external sphincters, same thing for defecating. These sphincters, you have a sphincter between your esophagus and your stomach, and between your stomach and your small intestines. These sphincters, these round sphincters, also how they operate controls organ volume. Controls organ volume. Do you have to think about when you're swallowing, coffee or whatever you're drinking or eating? Do you have to think about your esophageal sphincter to your stomach relaxing? No. Or when your stomach's done with whatever it's doing and the contents are going to go to the small intestines, you have to think about the pyloric sphincter emptying, opening. No. All under involuntary control, aren't they? But their functions, smooth muscle and sphincters, their functions are to control organ volume. Are we good with that? All right. So when we get to the muscle chapters and we're talking a little bit more about it, you'll have that much of a foundation with it. These muscles are very different than the other categories in that, and again, these have been stated for you. Skeletal muscle has really parallel looking fibers, and then you can see these nuclei are usually between these fibers here. And do y'all see these little mini, these are called striations. They're perpendicular little lines, wavy lines. Skeletal muscle, just nothing else looks like skeletal muscle except for that. And so anyway, but what muscles mostly are packed with protein, contractile and regulatory proteins. So they look very different from the other tissue cells that we saw. They look very different. And their main function is that they can contract. Now, certainly they also have elastin fibers inside of them that allow for them to go back to their original shape. But the main thing that's unique about a muscle is that it can contract. Okay? Now, if you look at the skeletal muscle, how parallel these fibers are. And by the way, what is steak? Does anybody like steak? So steak is what? It's muscle. Okay. So when you think about what steak looks like macroscopically, you know, you can kind of think about this. But anyway, this is skeletal muscle. Now, cardiac muscle is, if we got this in a higher magnification, we would still see some striations. These are the fibers, but the fibers look really branchy, don't they? You see how branchy the fibers look instead of the nice parallel? And then smooth muscle doesn't have, that's cardiac, excuse me. And smooth muscle, you can't see those fibers though, the striations. Smooth muscle is going to not have the striations, and it just looks smooth. It doesn't have those striations, so whatever. We're going to look at those in lab. We're going to look at 3 muscle types: cardiac, smooth, and skeletal. And all we're really going to have to do to get 100 on the quiz is to just say when we see it that it's muscle, right? Okay. Okay. If I were to go back right now and show you all compact bone and adipose and blood, they look very different in muscle. And then the epithelial are really tightly, you know, they have these little tight, they look like they're lining. And if something looks like it's lining a tube, what is it? Lining? It's epithelial, right? So the ones I give you, I promise, when y'all are looking at them, you're going to think, okay, they really do look different from each other. I can get this. Now, what I'd like to, I know y'all are probably, are y'all beat? Do y'all feel like somebody's beat you with a stick? Okay, I feel a little bit like that, and I didn't even have to take the test this morning. But guys, I do want to finish up a little of this, and there's just very little bit that I want you to be responsible for this. But we said that epithelial tissue, the cells do need to be tightly joined, right? And there are particular types of ways that they're joined. We know that proteins help cell membranes carry out functions, right? So we know that has a part to do with it. But the main thing I want you to know about this is there are different kinds of cell junctions. Some of them are going to allow for rapid diffusion from nutrients from one cell to the next. It's almost going to be like a little tunnel, right? Almost like that. Some of them are going to act like zippers. They're going to literally act like zippers. So if you zipped it up, you know, it's a pretty solid connection. Some of them are going to act like, you know, almost like, I don't know, even more tighter junctions. There's one I want you to know the name. It's called desmosomes. These desmosomes are a type of cell junction that some people have issues with these because there's an autoimmune disease that can attack these. Tell me again what an autoimmune disease is. It means your own, those white cells that I said are no joke. Remember those? We don't want those attacking our own cells, do we? So can you catch an autoimmune disease? No. Absolutely not. It is your own cells attacking your whatever specific body type. This is the one that I'm talking about is called pemphigus vulgaris, but there's other types of pemphigus. It's not just vulgaris. I think it's in the notes. Is it? What'd you say? Okay, what page is that on yours? It's in a deeper insight. It's called pemphigus vulgaris. But anyway, I really want you all to know about this autoimmune disease because, like most autoimmune diseases, I don't know if y'all noticed this amount about people that you know who have autoimmune diseases, but some people get them they're horrible, and they're compromising their life, the quality of their life, and the quantity. Like it may take them out of here early, right? And some people can have the same autoimmune disease, and you know it's annoying, and they come in and out of remission. You know, it might be for months and even years at a time. They have no trouble, and then they'll have a flare up, you know, or whatever. And then about a third of people, you know, really it doesn't bother them too much. This is one of those. This is like that a little bit, but I'll never forget an 18-year-old that I saw die from this. Pemphigus vulgaris. And if you can imagine that you've got somebody's cells that are attacking the junctions that are holding their cells together. This is like if you've ever imagined a burn, you know, burn patients lose their skin, don't they? They can lose whole huge areas of their skin and they're weeping fluids. But instead of being burned from the outside, this is burning up from the inside out. Now, most people who get this are going to have like maybe some little vesicle blisters on certain parts of their body. Might be in their throat, mouth and throat. It might be on the back. It might be on their trunk, you know, and they'll come and go and they'll be annoying. But this young girl came in and they thought, and it sort of confused the issue, because she had just been put on an antibiotic for a urinary tract infection. 18-year-old with urinary tract infection is really common, right? So she got put on an antibiotic and she comes into the emergency room at UVA, and she's got, it's more than hives. It's more than systemic hives. It's looking really rough, right? And so immediately thinking what? Reaction to the antibiotic, right? And so immediately, you know, starting to try to reverse this allergic reaction, but it wasn't. It was this, and it came on suddenly, and she got put in within the first 24 hours. She was put in birth unit, but in 2 weeks she was gone. She did not make it. We just could not stop her immune system. Was attacking her cells, and literally she was burning up from the inside out. Now I have never seen a case like that since then, and I've actually diagnosed somebody, but it was somebody who just had, she said she'd been going for several years, and they just didn't know what were causing these little vesicles that she had, and anyway, that's what they were. And so usually it's just mild and annoyance, right? And you can come in and out of remission. But this is not great, Pemphigus vulgaris, and there's different types of it. Yes?
How would you repair damage from that?
Well, if you can stop the attack, it will repair just like blisters would, because they look like tiny blisters. That's what they can look like. Now, don't go home if somebody's got some tiny blisters, because they may have chiggers. You know, I don't know what they have. So, you know, a lot of things can look. I feel sorry for dermatologists. I tell people this when we get to the skin chapter. I feel so sorry for dermatologists because there's so many different rashes that can look. A rash can look this, but it could be 100 different diagnoses. You know what I mean? So this is why I said about mamas telling you that you had chicken pox, but it might not have been. You know, there's so many things that can give those kinds of like vesicular rashes. But anyway.
So the UTI was causing the autosomal?
No, that was just another thing that was going on. She had a UTI.
So what caused all of a sudden after 18 years?
So people don't know why people develop autoimmune diseases. We do think there's a familial component,
right?
But it doesn't have to be. No, that the whole being put on the antibiotic thing was just not even part of the issue. It was that she had developed this, and it was acute, and it happened, and it just went really fast. So, but anyway, our cells are, for the most part, those junctions
Come out as blisters.
Little tiny kind of blisters, and yeah, it'll be that's how it kind of don't be paranoid, okay? The worst people about hypochondria are nurses and medical students at first, because everything that you hear about, you think, I have that. I know I have that. Especially parasitology, I did, because I grew up on a farm, so I stayed barefoot the first 18 years of my life, you know, in fields and cow pastures. And so when I got to a parasitology class where you can get, you know, all of these parasites from swimming in creeks and ponds and going barefoot in fields, I was like, oh my God, I know I had that one. I know I've had that one. You know, And then you learn that most people, they're just a part of your friends if you have them, and they're self-limiting. So it's okay to have worms, as long as you have a good immune system, you're okay. Anyway, glands. So I want you to know glands secrete. Okay, glands secrete, and they may be these glands secrete. They may be endocrine or exocrine in their function. So exo means what is a prefix? Exiting. And endo means it's going to stay where? Within. So exocrine glands have ducts, because these glands are going to be secreting into ducts, and those ducts would eventually exit, wouldn't they? So sweat, mammary, tear glands, these are all secreting products, aren't they? Salivary. But endocrine are going to be releasing their products that stay in the system. So they're usually going to be secreting their products into the bloodstream. So is an important function of blood delivering hormones. Yeah, it really is. That's another function of blood, isn't it? So these hormones are molecules that will cause a reaction in their target cells. That makes sense? So hormones are simply these molecules that end up causing reactions in their target cell sites. Some hormones have widespread effects. So many body cells can be affected by the hormone. But some hormones are very, very specific, and they're only going to have a certain target site that they go to. I had talked to you all about insulin is a hormone, isn't it? And its target site is going to be many cells, because do most of our cells need glucose for aerobic respiration? Yep. So, right. But then also, I'm trying to think of one that you all heard of. But then we also have a hormone. Have y'all ever heard of prolactin? Yes. Prolactin is a hormone that targets the mammary tissues to make what? Milk. That's right. So that's a very specific target, isn't it? Prolactin. So we have these hormones that get secreted by these endocrine glands, and if they are secreted into the bloodstream, then we refer to them as the endocrine glands. Exocrine, it's going to be exiting. Okay, that's fine. The histology of these, this is FYI, which means what? I'm not going to test you on it. I just want you to know, would it even make sense that all the ducts would look exactly alike? No, that wouldn't even make sense. So some of them, these are like the mammary and the pancreas. So they have these different looks. Some of them are going to secrete by different ways too, through exocytosis, like taking something and sending it out into the duct. Do y'all see that? What is this space here called? Lumen. And if the cells are tightly lining, these are epithelial, right? And they're secreting. So that's exactly right. Some of them are going to secrete by actually undergoing mitosis, getting further away from their blood source, and then they start to what? Die. And when they die, they release their products. Right? That's just FYI. Just FYI for you for that. And then I don't think, let's see. This part is going to be something we want. As far as tissue goes, just a couple of terms. Again, are there any such thing as big words? No. They're just, no. So I want you to know that hyperplasia means tissue growth through cells multiplying. So there's going to be more, tissue's going to be growing through cells multiplying. Hypertrophy though, is going to be enlargement of those pre-existing cells. So if y'all have ever seen somebody bodybuilding and they look like their muscles are getting, whatever, they're not getting more muscle fibers and cells. That's not what they're doing. What's happening? They're not making more muscle cells. The cells are getting bigger. Yeah, the fibers are getting bigger. So that's what's happening here. Neoplasia is development of a tumor. So it might be benign, might be malignant. Do we know the difference between those two terms? Yep. Benign means it's encapsulated, localized, staying in place, right? Where malignant means it's not encapsulated and it could metastasize. Right. And we know what differentiation is, don't we? All right. Now, guys, I need to tell you all this word, and I need to warn you about this word. Metaplasia is a term that most in health sciences, you don't want to hear that term. Because if you hear metaplasia, typically it's not indicating anything good that's going on. But it happens occasionally in development. It happens normally and naturally in development for some things. Like I had talked to you about simple tissue lining the vagina before puberty, didn't I? So should the vagina be receiving anything before puberty? No, it's got a simple layer lining it. No, it should not. But after, and puberty actually happens before the first menstrual flow. It's usually, this is starting a couple of years before because things are going on already before the first menstrual flow, right? So this stratification is happening. So that's called metaplasia. When one tissue changes its structure and function. So that's what metaplasia is. But when you all hear about it in health sciences, it means that this is bad news because what do you think is really happening usually? What do you think is usually happening? Precancer or cancerous conditions. Precancer or cancerous conditions are happening. So that's how you usually hear that term. But on rare occasions, it's just, y'all already know what stem cells are, don't you? Don't you? Yes. Okay. What? Does anybody know? Everybody knows that the stem cell is an early immature cell. The earlier the cell, the more potential that the cell can become what you need, right? So, right. But the further along down the development differentiation line, then it's going to be dictated what it can become, right? Right. Do adults have adult stem cells? Absolutely. Adults have stem cells. And, you know, these terms, when you think about how potent they are, multipotent versus unipotent, potent, then, you know, it just means how much use would this stem cell be? What could this stem cell become? If it's an early stem cell, then it has more potential, doesn't it? Because it hasn't differentiated to a point where certain parts of the DNA are cut off. All right, so I think I had talked to you all about that. If you have any questions, I'm not going to make you go into that much of it. Let's start this when we get back, okay? I think we're not going to do lab today. So we'll do it Thursday, and we're going to have, so we are going to expect to be here at least until like 1 or so on Thursday, okay, 1:30 maybe, all right? But I think this is enough for today. Do you guys have enough in your notes for a study, guys? Yes. Let me ask you something. Are you already kind of sensing, was this already a little bit more comfortable than chemistry?
Yeah.
Yes.
And it's going to get even more so. All right?
So on Thursday, will we have chapter 4 lecture quiz, or how is the chapter 4?
Oh God.
I didn't want to be surprised.
You don't want to be surprised. Hey guys, since truly like next week we'll have a test on 4, this is 5, and 6. You really do need to review. Why don't you review 4? Because you haven't had a lecture quiz on chapter 4, have you?
No.
Okay, so I'm saying this, and that y'all can thank Nicole for this. You guys, because that was important, bring that up. You all really review chapter 4, and we'll have our lecture quiz on chapter 4. I won't include today's, Thursday, and then Thursday we'll just finish up the last few little notes on this and start skin. And you all can then review that for the next Tuesday, okay? But I'm telling you this, and y'all can hold me to it if I forget. But on Thursday, your lecture quiz will be strictly from lectured notes on chapter 4. Fair? And thank you, Nicole, because that was important. Because I don't want people not looking at chapter 4. And I know people probably didn't get to look at chapter 4 because you were studying for this test. Is that right?